Hello everyone. Today I would like to present what is epigenetics and what is its role in maternal health. The reason why we talk about this is first of all giving the general outline of this talk. Today we learn about what is epigenetics. Then we look at what is the role of epigenetics in maternal health, and I'll also talk about the concept of fetal programming, which becomes extremely central towards our understanding of epigenetics in maternal health. Then we look into the transgenerational implications of what is role of epigenetic regulation, and finally we'll talk about some clinical and public health significance of epigenetics. So throughout the course of today's discussion, we are going to this is like the most important molecule for today's talk, and this is not just the double helix of the DNA as you see on the left hand side, but towards the middle and the other side you can see that this particular structure is wrapped into a very fine intricate structure so this is we call as the common thread of life. This is the chromatin molecule. So I'm going to talk about this and what is its role in epigenetic regulation. So the talk is divided into three parts. So this is the overview of the talk, and what we'll first understand is, as I said, again, epigenetics. I'll give a historical perspective and also talk about some of the modern advancements in this field. And then finally, we talk about the cellular inheritance versus organismal inheritance. So then we'll understand about the human physiology and again the role of epigenetics. And then I'll take a single story about one vitamin, which is vitamin B12, which plays a central role in epigenetic inheritance. And with this illustration, I guess we'll be able to understand what is the role of epigenetic regulation in human health. So as I said, initially I'll provide a genomic context just to set the base for understanding what is epigenetics, because I guess many of us are aware of genomes, genomic sciences, because of the talk about the entire Human Genome Project. So the picture that you see here actually is quite an interesting picture, because what you see here in this bookshelf are different volumes, and these volumes are numbered as per the chromosomes, the units of inheritance. These are numbered from 1 to 22, and then the X and Y. As you can see from the numbers, these are the numbers of the chromosomes. So human cells have 23 pairs of chromosomes, and what you see here is a single printed copy of our genome. So which means we figuratively and literally carry so much of information in each cell of our body. If you go close and open one of the books like this and read the information reads like this, which is a string of 4 alphabets, A, T, C, G, which are the building blocks of our genomic material. And you can see that there is no punctuation here. It's a contiguous set of instructions encoded in these 4 alphabets. So obviously, what are genes then? If this is our genome, collection of our genetic material, all the chromosomes put together, and if it reads like this, then how do we define a gene? So what I have done here is taken an illustration, one of the genes, which is actually one of the smallest genes in the human genome, and the sequence is here. Okay, so as you can see, a string of 4 alphabets, and there are thousands of these nucleotides, or the alphabets as I call it, and you can see that it's very difficult to discern any pattern in this, but we call it a gene. So how do we know that this is a gene? Okay, so in the next slide, what I have done is, of course, this summarizes the knowledge we have accumulated over the past several decades using molecular biology, genetics, and multiple other tools to understand the function of genes. And what you see here is the same gene now decorated in different colors. Okay, so this is actually the genetic code. And what you see here in different colors are different elements in the genomic sequence in the code of alphabets. Now you can see that there are some in different colors. The blue, the yellow, the red, and all other colors indicate different genetic elements in this gene. So obviously the gene is divided into multiple different parts, and some of them, like those in the green color here, might be encoding what is called as the protein, which are the workhorses of the cell. So the DNA is encoded into RNA, and RNA then gets translated into protein, and proteins are the workhorses of the cell because they do the work in terms of machines. So they are different types of proteins, which we'll come to, but this in a sense is the genetic code that defines how the genes function. But then ultimately what we need to understand is this enough? Is this knowledge of the genetic code enough for us to understand the complete functioning of this particular gene or other genes? So does this really provide a complete picture? So that's the question that we're going to address today. And the answer is, I'll just give you the answer right up front, that indeed knowing the sequence alone or the genetic elements in that alone is not necessarily enough to understand in toto the complete function of any gene for that matter. So then what else is required? Now the question is, does it really matter how many genes we have? Okay, and I'll come to it why we have addressed this question. The answer to this question is almost meaningless these days after the genome sequences for large number of species have been elucidated. So the reason being that each gene can give rise to several proteins. Okay, so as I told you, genes can encode proteins, and there are ways by which through the split gene function, genes can encode multiple proteins through alternative splicing. Each protein can be modified in multiple ways, what we call as post-translational modifications. Again, I will talk about these. These are very small ways in which the properties of the proteins can be modified by attaching chemical entities. And then finally, these modified proteins can further work in complexes. For example, if you have three proteins A, B, C, A can partner with B, B can partner with C, A, B, C all can come together. So there are multiple permutations and combinations in which these proteins can work together, and each of them will have a distinctive function. So therefore, with just a few genes, one can really encode multiple complex paradigms. And that's why it really doesn't matter how many genes we have. And because of this, now scientists have come to appreciate that genome size and organismal complexity are not directly correlated. And the reason I put forward this is to understand why it is important to go think about beyond the genome itself. There are genomes of multiple organisms that have been characterized, and it has been very clearly seen that the size of the genome does not necessarily give rise to the various complex phenotypes of an organism, the way the organism actually functions. I can give an example that some of the multicellular organisms, including humans, we consider humans to be most evolved species, and these necessarily do not have the largest of the genomes. Many plant species, many single cell eukaryotes, including amoebae, have much larger genomes than that of the human genome. Then really does it matter what is the size of the genome and the complexity? Because the complexity, we believe, is not necessarily dictated by the size of the genome or the number of genes. Even the fruit fly, which is again used as a very favorite model for developmental and genetic experiments, has number of genes which are very close to what the humans have. It's not like humans have twice or thrice the number of genes as compared to the fruit fly. So which indicates that indeed we need to look beyond the genes and the genomes itself to understand how they encode complexity when it comes to organisms. And that's what epigenetics is actually about. So let me now introduce another term. As I mentioned, chromatin, which is the molecule shown here. It's a molecule of DNA in complex with proteins, and these proteins are highly basic. Okay, so nature has smartly evolved this scheme where DNA, which is acidic because it's deoxyribonucleic acid, makes very tight complex with an oppositely charged molecule. So the basic proteins, which are histones, and these proteins which are histone as well as other proteins which are highly positively charged make very tight bond with negatively charged DNA, and as a result of that they make this beautiful complex, what is called as the nucleosome. And this is the fundamental building block of chromatin. It's a highly dynamic material which carries large amount of epigenetic information, and the flags that are shown here are essentially the epigenetic information that is encoded in chromatin. So we'll talk about how do these flags or the epigenetic information contribute to the function of the genes. And finally, cells in the organism in a single organism carry same genetic information. For example, cells in different organs of our body from head to toe are all identical in terms of genetic information, barring some exceptions. For example, T cells which have some recombined elements, and of course RBCs which do not have nuclei at all. But then other than that, each cell carries identical genetic material, but most importantly they look different and they express different types of genes. That's why cells in our brain function like neurons, whereas cells in our muscles function like muscles, and they express different types of proteins and of course have different functions. So how does it happen despite having identical genetic information? Okay, so that is the question we are going to address. Let us then understand what is epigenetics, and this is a term that was coined long back, way before the structure of the DNA itself was deduced, and that is by Conrad Waddington, a British geneticist. And what he said was, he coined this term by fusing two terms. One is genesis, generation, something to do with genetics, which we all know is the science of heredity. How traits are transmitted from one generation to the next is the science of genetics. But what is epi, what is beyond and above that of genetics, is what we are going to discuss today, and that's precisely what he defined as epigenetics. It is the branch of biology which studies the causal interactions between genes and their products, which bring the phenotype into being. Phenotype is what we see. Okay, so my phenotype is this. Phenotype of the screen is what you see here. So this is what we see. This is how an organism is built. But that is happening because of something intrinsic. The genes essentially make me, right? So, or everybody else. So that is what is the genotype. So genes or the genotype gives rise to the phenotype, which is what you see outside. But the transition from genotype to phenotype is the most complicated transition in nature, and that's exactly what we have now tried to understand through an epigenetics lens, and which is the beauty of what we are able to now understand in modern biology. The other words, heritable changes, which is genetics, in gene expression, which are not attributable to nucleotide sequence variation, means the alphabets that I talked about when I showed you the genes is essentially epigenetics. So if there is a change in the alphabet, that is called the mutation, which are many times quite deleterious. Mutations can cause diseases like cancer and many others. So obviously those are not necessarily good for us. So what we are talking here are changes where there is no difference in the nucleotide sequence. They remain identical, but something else happened, and that is the epi part of it, which we are going to talk about. So how does nature bring this upon in the cell? Okay, so there are tools of epigenetics. So there are 3 tools of epigenetics, and these are quite simply categorized as what the images you see here. So there are writers, there are erasers, and there are readers, which enable exactly what these words mean. So there are a set of tools which are enzymes or protein molecules which can write the epigenetic code or the information. There are proteins which erase that information, and there are proteins that allow reading of that information that writers have added onto DNA or proteins. So essentially these 3 tool sets is what epigenetics is about. So we try to understand the function of writers, erasers, and readers, so that through this we'll understand how the entire epigenetic regulation is brought about in any biological system. And even today we're going to talk about some of these. Once you understand the function of these 3 different types of subsets of tools of epigenetics, essentially we are able to follow how do they function in coordination in any biological system to bring about the transition from genotype to phenotype, from genes to that of the being. And this is essentially, in a nutshell, what is epigenetics about. So let's dive deep into what does it do to the genomes. So here I have taken a paragraph from a text, literally copied it verbatim, and this again is paragraph, something related to epigenetics. So if I were to, let's say, read this, and I want to just remember something important here, I would highlight it, right? I will use maybe yellow highlighter, or if I give it to someone else, that person might use something else to underline a sentence or a word, or you put some marks on the side just to say that, okay, this part of the information is important for me. The tools of epigenetics do exactly the same thing to the genomic information. So let's say this is the genomic information, these are the alphabets, and epigenome or the epigenetic tools do something like this to the same information. I have highlighted this in yellow, somebody else highlighted something else in orange, and this word has been underlined in a different way. So indicating that at different times, some different words or different contexts become more meaningful for that system. Let's say these are 3 different cell types, identical genetic information, but it is utilized in different manner, so that this becomes more important in cell type A, this becomes important in cell type B, this is important in cell type C, right? So this is how, through identical genome, different information is extracted at different times in a living cell, so that the cell can bring a different phenotype into being. The cell can actually change into different, so that's the plasticity I'm talking about. So this is something that Waddington proposed in 1942, again, before even we knew about all of these genomic information. So he proposed that a cell, which is like a stem cell, can change into different cell states, different types of cells, which we now call as differentiated cells. Stem cells are precursors of all of these cells. So depending upon which terrain they move to, imagine a ball rolling down a bunch of valleys, and then finally it will land either on that side or this side, and based on that, its phenotype again will change. From an identical genotype, different phenotypes can be made using this analogy. So now, of course, we have understood this in a much better way, which I will show you, but this happens primarily through epigenetic modifications. So these modifications can be at the level of DNA, which is shown here. So this is the DNA methylation machinery, and what happens is that the regulatory sequences in the upstream of genes, I showed you the genetic code. So this is the start site of a gene to make an RNA molecule. So this region can be unmethylated or methylated. So the lollipops that you see here essentially show that the CpG dinucleotides are methylated, and based on that, the gene then gets inactivated, right? So you have a molecular switch now. Gene can be active or inactive just based on the methylation signature of the gene. So this is at the level of DNA, and the enzymes that do this the cycle is shown on the left hand side, how the CpG dinucleotide can be methylated through the action of these writers and erasers, and essentially that's what. So you add only one small group, which is methyl group CH3, to this cytosine, and that can completely change its chemical properties. Different reader proteins can recognize this mark and come and bind to that, and that's how the information is extracted at a biological level. DNA methylation happens to be a very, very stable mark, and it can persist throughout generations. People have seen that it can be carried through different generations. This process is called imprinting. We receive a set of imprinted genes from our parents, from the father as well as from the mother, and they do remember where they come from. The imprinted genes remember the parent of origin, whether they're coming from maternal allele or the paternal allele. So it's very interesting to see how genes also remember. They have an epigenetic memory of where they come from, So it's not like a random combination. They precisely remember and have an epigenetic memory. And again, that's a very interesting topic to talk about. The second modification is at the level of proteins, not at the level of DNA, and this modification is for chromatin. So these proteins, which are histones, which are the ones that make the nucleosome core particle, you can see that they have this beautiful tails which are projected outwards, and these tails have these amino acids, which are again their building blocks, get modified in so many different ways. Like there are 4 modifications shown here. Now we know that there are at least several hundred different kinds of modifications. It could be methylation, acetylation, ubiquitination, phosphorylation. Again, very small chemical groups that are attached covalently to the protein residues, and these are the ones that carry the epigenetic mark. Unlike DNA methylation, the protein modifications are relatively transient. Okay, so the histone modifications are also transient, and because of which they are regulated much more dynamically. Constantly they are added and erased, and also read in multiple ways. And you can just imagine the number of permutations and combinations in which one particular gene can be regulated, because each amino acid here in this tail is modified in a different way, and that's what gives rise to a different output, which indicates that indeed all of this can give rise to multiple layers of regulation of genomic information, and that's the beauty of epigenetic regulation. So coming back to this now, I can talk about how this is done in much more detail, but today we'll move on further because I want to talk about the Waddington's hypothesis. So here you can see now this is an animation, very nice animation to show how stem cells initially divide. They're all alike, but along with the epigenetic landscape of Waddington, now this is an animation where you see that these start moving down the valleys, and then eventually they change the colors, and finally when they end into the pits, when they go to the end of this process, you will see that they have acquired not just different colors but also different shapes. Okay, and these are the terminally differentiated cells, and you can beautifully see that the neurons look very different than the blood cells on the left. So imagine they all started alike, but at the end they have become completely different from each other, which indicates the phenotypic heterogeneity. How many different types of cell types are possible? In human body, there are more than 200 different types of cells, all of which came from the single cell. Okay, we start our life as a single cell, fertilized embryo, and after that it divides, and eventually each cell becomes different from each other despite having identical genome, and that is possible because of all these epigenomic regulations, which are shown in different colors here symbolically, but actually that is a series of permutations and combinations of epigenetic modifications, both at the level of DNA, proteins, and now we also know that it's also at the level of RNA. So there are 3 different ways in which epigenetic modifications can be done. And as a result of that, epigenetics influences multiple cellular processes. Now people have characterized this in a number of different paradigms, from stem cell function and maintenance to cancer, different diseases, as well as different differentiated cell types, and aging, which is also becoming an important problem now. To address aging and longevity, people are trying to understand how we can use epigenetic therapies. And of course, the diseases, diabetes and many other disorders like cardiovascular disorders are regulated indeed by the epigenetic mechanisms in addition to genomic mechanisms. So for every disease, there could be a genetic basis. Like for cancer, obviously some cancers are primarily determined by the genetic predisposition, but then beyond that, the second and the third insult could be epigenomic, and of course that comes from the dialogue of genes with the environment. So I will again talk about that, how our environment speaks to the genes and how that brings about different modes of regulation. And because of this, epigenetics is considered as probably the most important revolution in modern biology in the past few decades. And if you see the cover page of many of these magazines, you can see the titles. Time magazine came up with this, "Why Your DNA Isn't Your Destiny?" Or here in Outlook, in the Newsweek, you can see that you see a picture of a fetus And probably that's where the health begins, very early in life, in the womb of the mother. And this is precisely the focus of my talk from this point onwards. We are going to focus on this window of time because this is the most important window in which a large number of epigenetic modifications can happen, something that we call as a fetal programming, because this is exactly the place where the child's future, the life of the child and the health status can be determined. Of course, people have done large number of studies with twins because they have identical genomes, 100% identical genomes, but yet they could look different, they could behave differently because of their epigenomic modifications, because of the influence of the environments in which they have been brought up. So I'll give you a few examples of how drastically phenotype can change with the various other aspects in terms of the interaction with the environment. One example comes from these social bees, the honeybees, and you can see that this is a picture of a beehive, and there are multiple different types of bees, queen bee, workers, etc. So you can see that these queen bee, there is only one per hive, but there are thousands of workers and there are thousands of foragers, and these two look quite different and they also have different functions. Okay, worker bees of course do a lot of work, fetch nectar, make honey. Queen bee only lays eggs. There's only one per hive, and if you remove the queen bee, then one of the workers changes into a queen bee and starts laying egg. Okay, so that's a complete transition of the phenotype. I'll give you another example of a phenotypic transition. So here we are talking about army ants. Now look at the worker bee. You can see here at the bottom, and then I'm going to show you a picture of the queen of the same species with 100% identical genome, and that looks like this. It's a dramatic change, right? So look at the two sizes and they are essentially derived from the same genome. So how can two different phenotypes emerge from the same genome? And obviously that has an epigenetic basis. It's because of the influence of the environment and the behavior and everything that feeds into the genomic regulation. So I'm going to give you some examples of how environment influences susceptibility to NCDs, which are the non-communicable disorders, which are on the rise these days, which are different diseases like cardiovascular, diabetes. Of course, aging and many others are part of it. So one example is from the early life experiences. This also have implication in mental health. So these are some of the human cases, and we then create animal models to study those. So for example, in the postnatal environment, as soon as the child is born, the child can sometimes go through abuse, neglect, trauma of variety of kinds, and that can set lots of things in the body of the child. And people have studied what effects it causes on the behavior as well as the cellular and molecular changes at the level of the brain. But obviously to do experiments, you have to have parallel models in the animals. So similar models have been established in animals. These are rodents, to study the effect of the postnatal environment by looking at the inherent differences in maternal care. There is a model called maternal separation, which I'm going to show you, and then postnatal handling. So in the maternal separation model, what is done is the pups are removed from the mother. So this is the litter, and the pups are then removed away from the mother for 3 hours in a day, and then you basically do this for the first few days of the life of this entire litter, and then you look at the effects later in life. Okay, so assuming that mice have only about 2 years of life on an average or less, so you can basically see this early effects in the early life and the effect later in the adulthood. So that's exactly what has been done by a number of labs. We have also done some studies in collaboration with investigators at TIFR. So basically what you're looking are effects of early life adversities and this experience, how it regulates the epigenetics. So this was done at multiple levels. I'm just going to summarize it here because it's published in this article, and you can see that it changes cognitive features, changes some of the neuronal features, of course molecular changes which we talk about, and at the level of epigenetic changes. So you can clearly see that early in life the epigenetic modifications are different than in the late life. Okay, and that is exactly opposite of what is seen in the activity of the gene. So this is the name of the gene, which is a very important gene for behavior, regulating behavior, and you can see that it's exactly opposite early in life for the first 1 or 2 months in life, and then in the adulthood it switches. So epigenetic changes can also regulate activity of genes in a time dependent fashion across the lifespan. And then the most important experiment here was, can this be reversed? Because if it's epigenetic, you have readers, you have writers and erasers, so you should be able to erase this information. So indeed behaviorally if you can intervene, or if you provide drugs. So in this case, one antidepressant drug was provided to the animals, and that drug treatment, okay, shown here, also leads to erasure of these epigenetic signatures. Okay, so you can see in oral administration of the antidepressant leads to rewiring of epigenetic modifications in the brain of the animal. So there is a connection between all of this, which is quite useful to see, because many of these then can become part of the epigenetic therapies for mental health problems such as depression, anxiety, stress related disorders. So as a result of this, there are a number of papers. I'm just going to show you the headlines, which show that environmental factors can affect the phenotype through epigenetic changes within lifetime of an individual. So this is called epigenetic programming by maternal behavior. So we are actually studying the role of behavior at a molecular level to show how it changes the gene circuitry, which can give rise to a completely different phenotype for not just the same generation, but also the next generation. So that's the transgenerational aspects. And finally, I would like to also come to this aspect of the environment, which is diet, because what we eat is also part of our environment. It was external and then becomes internal, and then improper diets also can be risk factors for diseases. And this is becoming extremely important now because everybody has access to high calorie foods, the so-called fast foods, and many of them are actually not very good for our health. And how does it actually affect our epigenetic circuitry? So the epidemiological studies have associated specific foods with the incidence and severity of many non-communicable disorders such as diabetes, cardiovascular, and other chronic non-communicable diseases. But how does it work? So we have to understand at the level of epigenetics. So now there is a science which is called nutrigenomics, how diet influences gene expression. So whatever we eat, how does it affect the genes? Okay, so the diet affecting the epigenome and affecting the phenotype, which is what we are, finally leading to either a healthy state or a disease state. So we're going to understand all of this to understand how epigenetic changes can give rise to health or disease. And if we understand that properly, then we'll be able to devise therapies to avoid the diseases. And that's what is the important part of modern biology or healthcare. So this is one of the earliest examples of how diet can influence epigenetically something to do with health of an organism. So here, the study was done way back in 2003, Waterland and Giltl. They basically showed that provision of a methyl deficient diet, which is folate deficient diet essentially, which lacks methionine or B12. These are very small molecular weight compounds. This is a vitamin, and you can clearly see that the animals became obese, okay, just because through conception process the mothers were given deficient diet and it lacked a critical component that is part of the epigenetic cycle. So I'll come back to this, how it is important, and this was later on shown even in higher animals or in bigger mammals. So indicating that yes, indeed, this is an important aspect of gene regulation that can have a transgenerational implication. So that's why we now want to understand what are the transgenerational effects of epigenetics, because it is redefining almost everything in biology, and it's extremely important for the personalized healthcare and personalized medicine. So that brings me to the second part, from cellular inheritance to organismal inheritance. So what I mean here is that essentially from the molecular level, we talked about the epigenetic changes, the chromatin, to the cellular changes that gives rise to tissue level changes, which finally affect the organs, then the systems. This is the nervous system, for example, that affects different organs, and then the entire organism. Okay, so we are going from this entire path from molecules to cells to organs to tissues to systems to organism, and that's indeed what we need to understand in toto. Okay, and that's the systems level understanding of any phenomenon. So for epigenetics also, large number of laboratories are starting to understand this level of regulation. So we are again coming back to nutrition because that's the subject for today's talk. How do we understand the multi-generational effects of nutrition in health and disease states? So two examples I would like to give. One is an example way back from the Second World War, and this is a period in 1944, 1945. It was a situation imposed because of this unique issue that happened in German-occupied Netherlands, and it's called Dutch hunger winter in these years, 1944-45. And because of this imposed famine, that almost 4 million population was exposed to this man-made famine, because they were not provided proper nutrition, proper diets, because of the war situation, and they were basically put in a very small region and not allowed to have access to proper diet. And because of this, they had a very low calorie input every day. So the diet consisted of minimum like 400 to 800 calories per day. This is like a severe starvation that was imposed upon a very large population, millions of people. And then because of efforts of some scientists who actually followed up, they could study what happens to the offsprings of parents who have gone through this kind of a starvation period. Okay, so they studied children which were born or conceived rather during this period where the parents underwent starvation, and what happened to the children born to the parents that had the starvation. And you can clearly see, all these papers have shown, this is the published information, you can clearly see that these children had low birth weight, high obesity, which is again counterintuitive, right? Because the parents were starved in utero, the babies were also starved, but then in adulthood they became obese. And of course they had increased risk of type 2 diabetes and schizophrenia, again mental disorder. So it is amazing to see how diet can give rise to such profound influences across generations. Parents were starved and the babies had these effects just because their parents were starved. And you can see something similar in countries that had starvation for many other reasons, for whatever socio-political reasons. So one example is, of course, in countries like India, where for centuries the masses were undernourished. India has gone through several famines. And now, of course, the entire country has access to high-calorie diets almost everywhere across all the corners. So to recapitulate this, one of my collaborators, Dr. Anand Hardikar, created a cohort of animals which went through multi-generational undernutrition. And in fact, it was a really large study, lasted for 13 years. And for 50 generations, animals were given half the calories. And for 50 generations, they basically were deprived of half of the calories, and this essentially was tried to reverse by giving 2 generations of regular diet. Okay, nothing fortified, just back to regular diet as the controls. And then at the end of that, the rats were actually analyzed, and what was seen here is quite profound. Just because these rats had parents that were undernourished, they became completely. So I will not go into the parameters that were studied. A whole bunch of biochemistry and of course molecular studies were done, including epigenetic modifications. And you can clearly see these rats have become obese, the size. And if you now look into the scans of these animals, the white shows the fat deposition. And you can see that in the control, there is the normal level of fat. But look at the recuperated animals. There's fat from head to the tail. The entire body is full of fat. They did not eat more, they just ate regular food. But it's because their forefathers, their parents and grandparents were undernourished for multiple generations. These generations have become obese. So this indicates a thrifty phenotype. Your body will try to gather as many calories it can because it tries to remember the famine. And what if there is another famine? So the body remembers this and tries to accumulate all the calories into fat. And precisely this is what has happened in the Indian population largely. So that brings us to the last part of the talk, and this is the developmental origins of health and disease, DOHAD, which was a hypothesis put forth by David Barker at the University of Southampton. And he put forth this famous hypothesis, which is now being followed by multiple other labs, and we have also done some studies in collaboration with clinicians, the fetal and infant origins of adult disease. So much of the adult disease can be actually traced back, way back in utero, in the fetal stage, and that is what is the fetal programming. So what it is called as fetal origins of adult disease hypothesis is now called as the developmental origin of health and disease, suggests that the person's risk for developing chronic diseases in adulthood is influenced by events that occur during early development. And this early development is basically in utero. So what are the molecular mechanisms underlying this developmental origin of disease phenotype? So changes in DNA methylation have been associated with exposure to famine, as we saw. There are multiple examples starting from the Dutch hunger winter that I showed you. DNA methylation signatures change and cause defects in metabolism, and also can be sex specific. And there are a large number of such studies. So what provides the methyl donor to methylation reactions? So this is the last part of my talk, and I'll take one story, which is the example of vitamin B12, which is part of the one carbon cycle. So what is one carbon cycle? One carbon cycle is a chain of multiple biological pathways in which one carbon unit, basically, which is the methyl group, CH3, is moved across different molecules, and as a result, it basically is finally deposited in the epigenetic cycle. And that is done by these writers, okay, different proteins that can add this methyl group to the proteins or to the DNA molecules, and that leads to epigenetic regulation, okay. So all of this is part of the single carbon cycle, and you can very clearly see how the molecule gets transmitted from one to the next, and eventually SAM, which is derivative of methionine that contains this methyl group, And this cycle is actually in presence of the important cofactor, which is vitamin B12. And that's why B12 becomes important. And so that's why a randomized study was done. This was controlled trial called PRIA, which is done in Pune Rural Intervention in young adolescents, where they were given supplementation with vitamin B12 and of course protein and multi-micronutrients, and it was controlled trial. And in the newborns, the cord blood B12 and anthropometric measurements were done. But most importantly, we then looked at across these generations, and in these babies, the gene expression profiling changes and also epigenomic changes, which are important to understand the molecular mechanism. So I will not give the entire data, but look at the essence of what this data has told us. Are there changes in gene expression in cord blood in response to the micronutrient supplementation in the mother, which includes B12 as one of the enzymes or as one of the vitamins? And you can clearly see here, so this is the novel mechanism that we have proposed. So the SAM molecule, which donates the methyl group in the single carbon cycle to DNA or proteins, we showed that this entire cascade also upregulates the writers, which are the methyl transferases, okay, that add this methyl group to DNA or RNA or protein, and that's what actually causes the epigenetic regulation in this paradigm. So it's very amazing to see how small molecules which are taken through our environment, which is in this case diet, our nutrition, can contribute so effectively in a transgenerational fashion to our phenotype, to regulate whether we stay healthy or come down with a disease. And once we understand this, essentially we'll be able to provide therapies that can allow us to reverse this. And that's the beauty of understanding this entire science of epigenetics. So to summarize, I will give you 3 take-homes for today's talk. One is that environmental factors, including diet, have profound influence on gene expression patterns, which are governed by epigenetic modifications, including DNA methylation and histone modifications. Of course, RNA is part of it. I did not talk much about that today, but indeed these 3 molecules are extremely important. Secondly, the period of exposure or the treatment during the life of an individual plays a major role in determining the outcome of the diet-induced changes. The earlier it is, the more profound is the effect. So obviously that's why life in utero or periconceptional life is very important determinant of diet-induced changes and especially the health outcomes. But the best part of it is that it is reversible, because epigenetic changes can be reversed as opposed to genetic changes, because there is an eraser or there are molecules that can alter the activity of the readers, writers and erasers, and therefore reversibility of such changes can be exploited as a strategy for the therapeutic interventions targeting the epigenetic machinery. And that is the bottom line. That's really the most important part, where large number of scientists as well as different biotech companies are actually working on this to come up with molecules which are for the epigenetic therapy for addressing health issues in the healthcare segment as well as various diseases, so that we can come up with personalized therapies for tackling large number of non-communicable disorders. So I'll stop here by summarizing what I said, and this is a very beautiful cartoon drawn by one of my PhD students who is now graduated, Sneha. Drew this cartoon, and you can see here a pregnant woman, and the womb mother, in her womb you have this unborn fetus, and even more importantly you can see another fetus inside this fetus. So these are precursors of ovaries of the baby that will be out of this unborn fetus. So you can see 3 generations here. Okay, so this lady is the grandmother of this baby, and you can see that the effect of diet can be as profound across all of these generations. So therefore this is the window that we should target if we have to bring about any epigenetic change in terms of affecting the health of an individual. So I'll stop here by acknowledging the people, the funding agencies that supported the work in my lab, the Center of Excellence in Epigenetics, which started at IISA Pune, and now we have this unit at Shivanari University in Delhi NCR. And of course, I would like to thank my collaborators listed here, who have really helped in large number of these studies that I talked about. And of course, thanks to the Gangwal School of Medical Sciences and Technology for inviting me to give this lecture. Thank you.